Good day everyone. At the outset, I would like to thank the organizers for giving me the opportunity to present our work on dairy effluent conversion into biofertilizer using tailor-made microbial consortia. This is a work that my group has been doing for quite a few years now. Silence of spring haunts my generation. How do we get back the tinkle of a running brook? Clear water and rock phosphate may be the cause of the World War III. Our technology takes up the challenge of cleaning up the worst of the pollutant, that's the dairy effluent, with a minimalist operation using our friendly consortium of microbes. The biofertilizer byproduct promises lush green vegetative growth. Water scarcity is a major global crisis reported nationally as well as internationally. One of the major reasons of this depleting freshwater reserve is agriculture, which uses about 80 to 89% of the fresh water that we draw every day. It has been reported that by the year 2020, about 40% of the global population would be subjected to water stress, while 21 cities in India will run out of its groundwater reserves, threatening day-to-day activity as well as food security. One way to slow down this depletion would be to minimize the misuse of fresh water, if not stop it altogether, for purposes like agriculture. Selective microbial treatment of agricultural runoff, municipal sewage as well as effluent from food industry, followed by its reuse in agriculture for selected crops, is the waste to wealth conversion approach adopted by the microbial technology group. Now, the reason for selecting dairy industry was that it is one of the most polluting industries, which produces about 3 meter cube of wastewater per meter cube of milk that has been processed. This untreated effluent, if discharged into the water body, causes eutrophication. If discharged into soil, causes hypersalinity. The available technology are tedious and expensive, which utilizes about 1.5% of the annual turnover of any installation. Hence, it's crippling for the small and the middle scale installation, and an alternative for converting the plant nutrient rich pollutant, that's the dairy wastewater, into biofertilizer is sought. That would make the dairy effluent treatment affordable, sustainable as well as profitable. This is a representation of the 7-step process, what you see, which conventionally a dairy farm uses, and that makes the process lengthy and also makes it quite expensive. What my group has been working on is developing tailor-made microbial consortia which could selectively convert the nitrate pollutants that are there into ammonia and not convert it from nitrate into nitrogen gas. So, the nitrate is not lost as nitrogen gas, but under the C/N rich condition, as would be there in the dairy effluent, is converted into ammonia, which is very essential. Now, using this principle, what we have been able to do is, we have been able to develop microbial formulation after a couple of combinations that we have been able to do. So, using that microbial combination that we talked about, what we have been able to get is the final consortia which selectively converts the nitrogenous pollutants into a biofertilizer plant utilizable form. So, the technology description involves a microbial biofilm. As you can see here, the biofilms are there shown in the lower panel, a microbial biofilm which converts the nitrogenous and the phosphatic pollutants into a nitrogen and phosphorus rich biofertilizer. It is a single or a 2-unit operation of 16 hours with associated COD and BOD, which gets converted to plant utilizable form with minimal power consumption. And there is a complete reuse of treated effluent, replacing fresh water and chemical fertilizer used during cultivation, and it is associated with about 1.5-fold increase in yield of mung bean. The process is scalable and has already been scaled up to 9 meter cube per day capacity, and there is negligible dead mass generated. So, now the developmental status, as you can see, is that the consortia with well characterized microbes are there with us. Its biofilm formation on different matrices has already been worked out, so that once it is charged, it can go on working for a very long time. The process has been scaled up to 9 meter cube per day in a 5,900 liter reactor. The proof of concept has been established at Agartala, that is the Gomti Cooperative Milk Producers Union, with associated field trial, which has shown a 1.5-fold increase in yield of mung bean. There is also positive results seen in case of background, in case of Remy fiber, sorghum Sudan grass, potato, and currently the trial with maize is ongoing. Now, what is next? We have also further scaled it up to 7,300 liter reactor, which is at Bhubaneswar OMFED, and what you see here is the reactor arrangement, the charging process, and what is the treated effluent that comes out from here, which is directly used as a biofertilizer. Now, after this, what we have been able to achieve till now is a conversion from a lab scale, where the 1 liter reactor was running through a 27 liter biofilm reactor, then a 5,900 liter biofilm reactor, and finally the 7,300 liter biofilm reactor, where the process could be scaled with ease and with an efficiency which was maintained all throughout the operation. It was not only the production of ammonia which could be maintained with the scale up, but also the associated field trial showed an enhancement in yield, an enhancement which was even better than chemical fertilizer use for mung bean and also for black gram, as you can see here. So, our key resources are the microbial formulation that we have developed, optimized and even maintained with us, our technology and the technology know-how, the methodology for the complete solution that we have developed, the patent that we have filed, and our team, that includes myself, Shaun Rai Chaudhary, the chemical engineer in the group, Professor Lalit Mohan Gantayat, and the bioinformatician, Professor Ashok Ranjan Thakur. Now, the patent has been filed, as I mentioned, in India. We have improvised the process further, and we are in the process of filing the next patent, which would be an improvement over what is there, followed by the PCT filing. Our market would be the dairy effluent treatment plants, the agricultural farm adjacent to the dairy farms. Now, the value proposition, if we have to talk about it, we are talking about a process which is 6.5 to 7.5 times faster than the conventional method. It does not produce any solid sludge, and hence the removal of sludge, which is the main problem with industrial processes, does not occur here. The total volume of the plant is less than 25% of what would be there for a conventional ETP to treat that amount of effluent, compared to the 70 kilowatt of power consumption in a conventional system. This particular technology would involve just 6 to 7 kilowatt of power consumption for treating about 500 meter cube per day ETP effluent. Now, the process involves use of a tailor-made microbial consortia in a biofilm reactor for rapid conversion of the dairy pollutant into ammonia, which is a plant utilizable form of nitrogen. The liquid biofertilizer, supplemented with a plant growth nutrient through this biological activity, gives better yield than the chemical fertilizer. In addition, it cuts down on the requirement of fresh water as well as chemical fertilizer for agriculture. So, if we try to compare our technology with what is already available in the market, what we see is ours is a 1 or 2 step depending on the effluent nature, as compared to the 7 step in a conventional system. Our system requires 16 hours as compared to 105 to 120 hours in a conventional system. The power requirement is substantially less, 3 to 6 kilowatt as compared to 70 kilowatt in a conventional system. There is no water discharge in our case, while there is a copious amount of water that is discharged in a conventional system. There is no scum generated in our situation, and hence one does not need a scum treatment process. There is a value added product that is generated using our technology, which is not the case in case of a commercial system. It's a zero discharge technology and which sustains agriculture along with yield enhancement, unlike the conventional system. Hence, the small dairies can be compliant with the pollution control regulations without adding on the cost of processing the effluent in this high competitive market. So, this makes us feel that these unique advantages would make this particular technology more important or more desirable from the commercial point of view. Now, there are certain risks and barriers associated. One of the risks is that the consortia is very stable and continues to perform for an extended period of time. Next, we have to strategically design policies for preventing transfer of this technology to a third party, and that the performance needs optimum operational procedure. If I try to conclude what this technology has to offer, it converts the dairy waste water to a liquid biofertilizer within 16 hours in a single or a 2-unit operation with minimal energy requirement, and the entire water is used for irrigation. In case of mung bean, black gram, remi linen fiber, sorghum Sudan grass, the yield enhancement is between 1.5 to 2 folds as compared to the same crop treated with chemical fertilizer. This would make the process of effluent treatment self-sustaining through revenue generation while protecting the environment and ensuring green farming in future. So, I would like to thank my entire team, that's my mentors and my collaborators, Professor Gantayat, Professor Ashok Ranjan Thakur, the granting agencies, the Biotechnology Industrial Research Assistance Council, which has funded this project under the BIG scheme, and the Ministry of Human Resource Development, Government of India under the FAST scheme, the students and the staff, which include Ms. Nabunita Haldar, Ms. Mandakini Gogoi, Mr. Ajoy Modak and Mr. Usha Ranjan Bhomik. I am extremely thankful to the organizations, Center of Excellence in Environmental Technology and Management at Maulana Abul Kalam Azad University of Technology, West Bengal, Tripura University, where this work has been carried out in India, Technology Business Incubator at Kalinga Institute of Industrial Technology, India, where I am currently incubated. I am thankful to the dairy organizations who have been part of our study and have allowed us to carry out the work there. That's Gomti Cooperative Milk Producers Union, India, OMFIT, India, and Mother Dairy, India. Last but not the least, I am extremely thankful to Indian Institute of Technology, Kharagpur for this documentation facility. Thank you so much.